All right, welcome back Science 30s to our next lesson in Chapter 4. This is 4.3, and today we're going to be looking at meiosis. In the previous video, we looked at this cell cycle and we focused on kind of the overview of the cycle and then on mitosis, which was the replication of body cells and basically cloning body cells to grow and repair. But now we're going to look at this part of the cell where the body gets ready for sexual reproduction and makes haploid sex cells in the form of either eggs or sperm. And these are called gametes, and we're going to look at a little bit more complex process of meiosis compared to mitosis. Okay, so similar to the last video, we're going to have the starting or parent cell down there to show that we begin with what I'm going to call one green chromatid and then a red chromatid to represent a chromosome pair. Now that's a diploid cell, which means we have 23 chromosome pairs. That's how we're going to start. And now we want to make cells that have half of this, so combine with another sex cell or another gamete to make an offspring with a full set. So we need to get to half of this DNA. So we're actually going to go through those same phases of prophase, metaphase, anaphase, and telophase, but they're going to be a little bit different and we're going to go through them twice. So we're going to start with going through the cycles, calling it prophase 1, metaphase 1, and things like that. So let's take a look at the start. Now to begin, just like with mitosis, all the DNA is kind of in these random strands and they need to be duplicated and then condensed. Okay, I'm just going to skip that animation and jump to this point in prophase where it's all been condensed. So the condensing of DNA is prophase 1. And I've actually already skipped metaphase as well because metaphase is where the chromosomes are going to line up in the middle. So I've shown them condensed and lined up in the middle. That's the two first phases of prophase and metaphase. Now what's going to happen here in metaphase for meiosis compared to mitosis is quite different because we're going to have some crossing over of these chromosomes. So while they're there, they're kind of lined up quite near to each other. And then I'm going to show them kind of wiggling here, interacting with each other. And some part of one chromosome is going to switch or get mixed with the other one. So I've shown here as we kind of have the red has some green on it, the green has some red. Those have mixed to make new types of chromatids. This is a new combination of DNA that was not present in that original parent cell. This is actually a good thing because it's going to create a different type of offspring. It's going to have some different traits than the parent. Now you might think, why would that be good? Well, the idea is that if you have a variety of different offspring, there's more chances that if the environment changes, one of those offspring is going to survive. If they were all identical and there was a change in the environment that they couldn't withstand, they'd all be wiped out. But if we have some variety, some might do a little bit worse, but some might do a little bit better, and we have more chance of survival to the next generation. So that's why this part for variation and crossing over of genes is very important. Okay, so we've got through the first two phases. Now we're going to move on to the next phase where we're going to have the centromeres on the side are going to grab the chromosomes in the middle and pull them apart to each side. And this is called Anaphase, and this is the first time we're going through it. So Anaphase 1. Similar to what happened in mitosis, but what's different in this case is that we haven't just separated into two diploid cells. We can see that we started with a green and a red on that side, but now on these two sides of the cells, we have two greens over here and two reds over here. So we actually only have half of the DNA, but we've got a double copy. And then here we have half of the DNA and a double copy. So this isn't the same as just mitosis and splitting into two identical cells. This side and this side are different than our parent. So it's good to keep track of that. Now we are going to split this into two separate cells. So we're going to start to form that nuclear membrane in between, which is called telophase. And again, this is the first time this has happened in meiosis. So telophase 1. After that's completed, it's going to be into two separate cells, and we've completed the first phase of meiosis. So now we start round 2, and as it says at the start, we're in prophase 2 now because we're going to go through a very similar process. Now, I've already shown it here that the DNA would have been unclumped, and now it's got reclumped again into the chromosome pairs. Okay, so that's prophase 2, again, just the clumping of the DNA. And then the metaphase 2, which I've already kind of shown as well, is where it gets moved to the middle they're in the middle of those cells. So same kind of idea as the first round of phases. And now kind of same step for anaphase as well. We're going to pull this DNA apart. So the centromeres are going to grab on and they're going to pull these to their opposite sides. And now we can see that we have 4 chromatids, each looking a little bit unique. Now that we've kind of set them into those 4 chromatids, we're going to go into telophase 2, which is going to form that nuclear membrane, start to split into 2. And now we had our final stage where we have 4 daughter cells or 4 products from where we began. So it's a complicated process, goes through all those phases twice. But now let's look at what we have at the end. We began with a green and a red chromosome pair, okay, homologous chromosome pair. There was 23 of those. Now what we have as a result is we have 4 haploid cells, or ones that are not chromosome pairs, but just chromosomes on their own, or chromatids. They're also all different. We have the green ones here, but they've mixed in with some red, and the red ones there that have mixed in with some green. So we have 4 different haploid cells as our product. And this would represent either 4 different sperm cells or 4 different eggs, and they would combine with, again, the opposite, either sperm or egg, to create an offspring, match up with them, and then make a full set of DNA for the offspring. So I know that's a lot of information. I hope the animations helped, but let's put it into our notes now. Again, we have to start with the DNA being duplicated, and then in prophase 1, it's going to condense into those chromosomes. Then for metaphase 1, we're going to line up the chromosomes in the middle at the equator. And the important thing that happened in this metaphase 1 was that there was the crossing over or switching of some of that genetic material to create variation in the offspring. Then once they were lined up in the middle and crossed over, we pull them apart into separate sides and form the nuclear membrane in telophase to create 2 different cells. So we have cell division at the end here. Where we saw in the animation, it actually has haploid cell because it's only got half of what we started with. It's just got duplicated versions of that. So like 2 greens in one cell and 2 reds in the other cell. So now to split those 2 greens and the 2 reds up, we have to go into the next phase. So now we're going into prophase 2, which is where we're going to condense the DNA again into its chromosomes, line them up in the equator for metaphase, anaphase, pull them apart, and telophase, form the new nuclear membrane, and we end up at the end having our 4 haploid cells. So a lot to know there, but I'm going to summarize it with this last animation, or last kind of summary diagram, where we start with that parent cell, and that cell is then going to duplicate its DNA, and we can see the green pairs and the red pairs. Then it's going to do the crossing over in that metaphase 1. Then it's going to split into these haploid doubles. Remember, 2 greens and 2 reds that have the crossovers in them. And then those are going to get pulled apart into our 4 separate daughter cells. So hopefully that kind of sums it up a little bit. Again, doesn't show the details of the prophase, metaphase, anaphase, telophase in this, but I like to look at that diagram to see the overall process that's going on. So I hope that helps you to understand this concept. And that is it for this video on meiosis. Thank you so much for watching, and I will see you in the next video.